Anything you want, we got it right here in the OUP. Hi, this is Jim Islip with MSU Extension, and this is What's Up at UPREC on December 8, 2021. Today, the crew here at Chatham is processing CBD hemp plants as part of a research project.
Hello everyone, I'm James Dedecker with MSU UPREC. This week we're in our freestall dairy barn bucking CBD hemp. This is the process of removing the leaves and flowers from the hemp stems, and it's really a process that represents what growers would be doing if they're creating CBD biomass for the extraction market, meaning that the flowers and the leaves that contain the cannabinoids, mainly CBD, but other ones as well, would be going through this bucking process and then to an extractor that would either use chemistry, solvents, or heat and pressure to remove the cannabinoids and then get those into products for the marketplace. So this is our second year of CBD hemp research here at UPREC. We had a trial in the field with about 25 CBD hemp varieties, and starting in October, we began harvesting, and that was a matter of cutting the plants down, weighing them for a wet plant weight, and then we brought them into the barn to dry. So they've been in here for over a month now, drying, and pretty much got most of the moisture out of them. They should be down around probably 12–15% moisture at this point, but it does fluctuate somewhat just with the ambient conditions in here. The dairy barn actually is a really nice place to do this work. It's set up actually quite well for hemp, even though that wasn't its original intended purpose, of course. We've got a lot of open space to hang the plants on the stall structures here. We've also set up some additional wires for more space to hang plants. And we have also our exhaust fans that allow us to pull air through the barn and out of the barn to facilitate that drying process. So I'm going to talk through what the kind of bucking process looks like for us and a little bit about how that data will be used. So we start with a dried plant hanging, like you can see behind us here. And those plants, the first thing that we're going to do is weigh the whole plant. And that's because we want to get a dry whole plant weight so we can later understand harvest index, or what fraction is the harvestable portion of that plant. And that we've seen some differences in varieties on that metric alone in terms of how much leaves and buds are there relative to the stock material. So we take that whole plant, weigh it, and then it's going to go into the bucker. This is a piece of equipment from Capital Creations, is a small manufacturing company downstate in Eau Claire, Michigan, almost all the way down on the southern border of the state. And it's farmers that have developed this piece of equipment and their company. And all it does, again, is strip the leaves and flowers off, and it's set up kind of like a chicken plucker, where we've got two rollers on the inside, top and bottom, with fingers on them that are spinning in opposite directions to kind of suck in the plant and strip off the material that we want to capture. That material comes out the back end here, and it's caught in this barrel that sits there on the back of the bucking machine. And that is actually something that we added on. Our mechanic, Joe Charleboy, manufactured this portion because mostly commercial growers that are using this equipment are bucking into a tote or a super sack that would be, kind of their intermediate storage before marketing the product. But for us, we need to separate each plant. So we're using this bin to capture material from individual plants. That bin then comes over to the scale, and we're able to weigh the amount of biomass that comes off an individual plant. And then we're taking a grab sample as well, and that's getting weighed too. And that grab sample is serving two purposes in this case. One purpose is that it is a dry matter sample for us, so we're able to weigh it at ambient moisture here in the barn after it's collected. And then we're also going to be drying it in the oven and re-weighing it to look at moisture loss to come up with a moisture concentration for each plant that's in this barn, because we need to correct our yields, like for any crop work that we do, based on moisture of the material. That sample serves a second purpose, and that is for a final cannabinoid analysis in the lab over at Lake Superior State University. So this hemp project is funded by a NIFA Tribal Research Grant, and we've got a number of partners on the project, including Bay Mills Community College and Waishiki Bay Farm, Ziba Mijwang Farm with Little Traverse Bay Bands of Odawa Indians, and Saginaw Chippewa Tribal College. Those folks have all been growing plots of CBD hemp, and in some cases grain and fiber hemp, contributing to the research in that way. And then all of our materials going over to Lake Superior State University, Dr. Ben Southwell's lab, for a chemical analysis. So we'll take a sample, from the bucked material, and that will go for cannabinoid analysis. There's something interesting happening there, the reasoning as to why we're looking at cannabinoids in this final sample. For CBD hemp in the United States, regulation is happening based on the concentration of THC in the field prior to harvest. So the State Department of Agriculture, in the case of Michigan, is coming out to hemp growers' fields, collecting samples from the top buds of the plant and running that through their lab. And growers have to demonstrate that their hemp is below 0.3% THC. If the sample comes back above 0.3% THC, growers now, under the USDA final rule, have an opportunity to mitigate THC in their crop. And the primary way it seems that the industry is looking at mitigating THC is through dilution or blending. And this is the common approach that we use to mitigate quality issues in a lot of different crops, grain in particular, where we'll take poor quality grain, high quality grain, and blend it together to meet a standard. Well, in the case of hemp plants, you have different concentrations of THC and other cannabinoids in different parts of the plant. MDARD is coming out and sampling the area that they would expect to have the highest concentration to look at, the greatest potential for exceeding that THC threshold, and that's in the buds at the very top of the plant. When we strip all the biomass off of the plants, we're blending the high concentration buds with the lower concentration leaves and flowers at the lower portions of the plant. And in that way, we're going to end up with a mixed sample of biomass from that plant after the bucking process that, in all likelihood, will have a lower concentration of cannabinoids than the sample that was collected by the State Department of Agriculture, or us in the case of the research, in the field during the growing season. And because this approach to THC mitigation is rather new, there's not a lot of great data to understand the differences between a flower sample in the field and what you're likely to come up with a sort of aggregated biomass sample. Some little bit of data that I've seen from other states is looking like they're cutting THC by 1/3 or 1/2 in that aggregated sample relative to the flower samples. So that's going to be really important, because we did have a number of varieties in the trial this year that did exceed the threshold, particularly later in the maturity towards 7 weeks post-flowering. So we do have varieties here that for a commercial scenario would have to be mitigated in some way in order for that material to be marketed legally. So that's what we're up to here. In bucking our CBD hemp. We're hopefully going to be finishing up this week or early next week here at Chatham, and then we'll be hauling the bucking machine downstate to Ziba Mijwang Farm and Saginaw Chippewa Tribal College right after the new year, and bucking their hemp as well, and getting all that data together for our final report, which should come out sometime in January. This year we had some really large CBD plants, and our yields thus far from the plants that we have bucked is reflecting that success, if you will. So in the field we were weighing wet plants anywhere between 20 and 30 pounds, which were quite large, quite a bit larger than last year or some of the other locations that we've had this year. And what we're seeing here with the dry plants is strip biomass yields somewhere in the range of 2 to 3 pounds per plant. So really high numbers, which is great to see. However, there's other things to think about there. One thing is just the issue of handling these large plants. Right now, a lot of hand labor is being put towards CBD hemp production, and larger plants are a little bit more difficult to handle. So that is a factor. Of course, high yields are great, but we also have to look at the harvest index and the relative weight of the whole plant and the waste in the form of the stalk versus the yield of the flowers and the leaves that will go to extraction. So big plants, looking like big yields this year, which is nice to see, but weights aren't everything.
That's what's up at UPREC. Hope to see you again next time.
Winnisec, Kinchlow, Kenton, Newberry, White Pine, Wakefield, Watton, kind of scary. I've been everywhere, man.